This video is going to focus on cellular respiration, which is how glucose is broken into ATP that we can use, and the byproducts are water and carbon dioxide. Oxygen is needed for the most efficient version of cellular respiration. The plan for the unit is to describe the three stages of cellular respiration and how they are related, look at how the structure of the mitochondria help for cellular respiration to function, explain the significance of the electron transport system to respiration, and discuss the importance of oxygen to respiration and photosynthesis and how both of those are linked together. So as always, structure follows function is a major theme, and the mitochondria, which is the organelle that cell respiration takes place in, has an outer and inner membrane. And these are important because the inner membrane has the enzymes for the electron transport chain and for ATP to be made, and the matrix has the enzymes for the Krebs cycle. And the outer membrane is what determines what goes in and out of the mitochondria, so nothing that mitochondria can't break down does not go in to just take up space. So the basic stages of aerobic respiration are glycolysis, Krebs cycle, and electron transport system. And these are how aerobic respiration is broken up. And each stage involves a series of chemical reactions catalyzed by enzymes. It's very similar to photosynthesis in that at each stage there is oxidation and reduction, there's NADH and ATP involved. So before we move on to the intricacies of cell respiration, I want to discuss the different molecules that you're going to see appear in different stages. So the first molecule is ATP and ADP, which basically ATP is adenosine triphosphate and ADP is adenosine diphosphate, so with just one less phosphate. So if you see in the picture, you'll see the string of phosphates, and this is our energy carrying molecule. And so triphosphate has 3 phosphates, and then diphosphate has 2, so when you remove a phosphate, you give off energy, and this is how we actually use energy, and so therefore during cell respiration we need to make ATP so that then we can use it during our various reactions. The next one is NADH and NAD+. This might look familiar from photosynthesis. Photosynthesis uses NADPH instead of NADH. They're very, very similar, and same with FADH2 and FADH. These are both electron-carrying molecules. So you see in the picture you have NAD+, which is oxidized version, and then to reduce it, you add an electron and it becomes NADH, because with the electron you now have the ability to bond the hydrogen, and this is how we are going to be transporting energy from one cycle to the next in cell respiration so that we can eventually make ATP. So these are both used in the same manner in cell respiration. And then the final molecule is glucose, which you should be very familiar with. Your 6-carbon C6H12O6 in ring formation, and this is what we start off with in cell respiration, and we're slowly going to break it apart by taking electrons out and breaking it down and making our NADH and FADH2, which then we're going to translate into ATP. So these are all of the molecules that you'll see throughout each little transition, and so now you can see the picture of them, and when they actually get used, you can see how they were used. So the general pathway is you start off with glucose. Glucose goes through glycolysis that actually partially oxidizes glucose. And then after glycolysis, there's 2 options. When there's no oxygen present, you go through fermentation, which there are 2 possibilities. You have lactic acid and alcoholic fermentation. And if there is oxygen present, then you go through aerobic respiration, which ends with the Krebs cycle and the electron transport chain. So this just gives you the general pathway, and now we're going to get into the specifics and go over what glycolysis is, fermentation, and aerobic respiration, and what happens in each. So glycolysis is the partial breakdown of glucose. So you always start with glucose and end with what's known as pyruvate or pyruvic acid, and there are 2 molecules. There are 3 major steps in glycolysis. There's what's known as the energy investment, the cleavage of the glucose, and then energy generation. Basically saying energy investment means you have to put in energy to gain energy. So the energy investment stage is when ATP is put in to glucose and attached. And then the cleavage is simply breaking the 6-carbon compound up into 2, 3-carbon compounds. And then energy generation is when ATP is actually made and NADH. And the final outcome is 2 pyruvates. So to look at it more specifically, we have glucose. 2 ATP are put into glucose, and you end up with fructose 1,6-biphosphate. So glucose, when it rearranges, it becomes fructose. And then when you add the 2 ATP, what happens is the 2 ATP go in, 2 ADP come out, and the phosphates are attached, hence the biphosphate. So the biphosphate means the 2 phosphates are attached to the fructose. Then after that, the cleavage of this 6-carbon compound into 2 compounds known as PGAL. PGAL is just a 3-carbon compound, very simply. Then the next stage is when you're getting energy out, so you are oxidizing PGAL, meaning you're removing electrons, and making NADH and ATP. So you have 2 NADH and 4 ATP and pyruvate. So the final outcome of glucose, you end up with pyruvate. And then you also have 2 NADHs and 4 ATP. But the total energy outcome of glycolysis is actually only 2 ATP and 2 NADH, because you put 2 in, and you got 4 out, so your net outcome is actually only 2, because you used 2 at the beginning. So this shows that the molecules that are formed in glycolysis, you have pyruvate is 2, NADH is 2, and the net of ATP is 2. So now what actually happens is if there's oxygen, it goes through aerobic respiration, but if there's no oxygen, it goes through fermentation, or what's known as anaerobic respiration. And that's what we're actually going to discuss first. It is a very simple process, but unfortunately not very efficient. So this is where your cell can actually decide, and it basically decides if there's oxygen, then it goes through aerobic. But if you don't have enough oxygen going to your cells, if you've been exercising or your breathing is altered, then you go through fermentation. So fermentation is the simple process of further breaking down the pyruvate, and there are 2 forms, and it only occurs when oxygen is not available. And everything stays in the cytoplasm, or cytosol. It never enters the mitochondria. So fermentation is completely in the cytosol. All it really does is regenerate NAD+, so no more ATP is made, but remember 2 ATP are made in glycolysis, so it is very important to actually regenerate that NAD+, so that glycolysis can continue going. So if you see in the picture, glycolysis, you need the ADP to produce ATP, and you need the NAD+ to produce the NADH. So this will all work together. And then you also have the 2 versions of fermentation, lactic acid fermentation and alcoholic, and each one has a slight difference. So alcoholic fermentation is what yeast goes through, and it helps make bread and wine and beer and all sorts of things. But those are the main purposes for humans. So as you see in this picture, the top line is for glycolysis, where glucose is broken down into 2 pyruvate and ATP is made. Then what happens after that, if there's no oxygen in yeast, they go through alcoholic fermentation. The 2 pyruvate lose a carbon dioxide each, so 2 carbon dioxides are released to make 2 acetaldehydes, and then those get reduced by the NADH and form NAD+. So the NAD+ can get regenerated to continue the glycolysis, because if there was nothing after glycolysis, the 2 pyruvate would just sit there, and then glycolysis would run out of the NAD+, and would not be able to perform, and you wouldn't get those 2 ATP. So in the end, you end up with 2-ethanol, hence why it's called alcoholic fermentation. Lactic acid fermentation is what we actually go through as humans, and yogurt and cheese go through it. And you'll see you get the glycolysis on the top again, except this time you don't produce carbon dioxides, and you actually just reduce the 2 pyruvates, regenerating that NAD+, and produce 2 lactate or lactic acids, hence the name lactic acid fermentation. So the only main differences between lactic acid fermentation and alcoholic fermentation are the organisms that go through it, the final outcome of lactic acid versus alcohol or ethanol, and that alcoholic produce CO2. So this is your basic overall outcome, what goes in, what comes out without oxygen. So you have glycolysis, and then you either have lactic acid or alcoholic fermentation. So this just shows you the basics, puts it all in a nice little table format of what is put in, what is made, and then what's recycled. So the purple is recycled, green are your products, your ultimate products. So glycolysis, it makes your 2 ATP, and then why the 2 NADH and the 2 pyruvic acids are put in purple is because they're used in the next stage. So they are not in the overall outcome. So the overall make at the bottom, as you see, is 2 ATP, a lactic acid, and or ethanol, depending on which fermentation you went through. So this sums up all of glycolysis and fermentation. So basically, cell respiration without oxygen. It is not very efficient because you only make 2 ATP, but 2 ATP is better than 0. So this just gives you a review of the pathway that we started off in the beginning. You have glycolysis, and then you either have anaerobic or aerobic. With anaerobic, you have lactic acid fermentation and alcoholic fermentation. And with aerobic, you have the Krebs cycle or the electron transport chain. So hopefully you kind of get the anaerobic side of things. And now we're going to move to the aerobic side of things in our next video.